Bacteria are everywhere and they're incredibly adaptable. They live in the soil, water, and even inside our bodies. Some bacteria are helpful, like the ones in our gut that help us digest food. Others can cause diseases. One of the reasons bacteria are so successful is their ability to share genes. This process, called horizontal gene transfer, allows bacteria to quickly evolve and adapt to new environments. Imagine bacteria as tiny factories, constantly trading blueprints. This trading helps them survive and thrive. There are three main ways bacteria transfer genes: conjugation, transduction, and transformation. Understanding these processes helps us develop new antibiotics and combat bacterial infections. Let's explore these amazing bacterial superpowers. Conjugation is like bacteria sharing secrets through a direct line. Imagine two bacteria side by side. One bacterium, the donor, grows a tube called a pilus. Think of this pilus as a bridge connecting the two bacteria. The donor bacterium then sends a copy of its DNA through the pilus to the recipient bacterium. It's like passing a note with important information. This DNA might contain genes that make the recipient bacterium resistant to antibiotics. This process allows bacteria to share beneficial genes quickly, spreading antibiotic resistance among populations. Understanding conjugation helps us develop strategies to combat the spread of antibiotic-resistant bacteria. Viruses are tiny particles that infect cells, including bacteria. Sometimes during their life cycle, viruses can accidentally package a piece of bacterial DNA. Imagine the virus as a delivery truck and the bacterial DNA as a package mistakenly picked up. When this virus infects another bacterium, it delivers the bacterial DNA along with its own. This process is called transduction. The recipient bacterium now has new genetic information from the previous bacterial host. Transduction can transfer various genes, including those responsible for antibiotic resistance or toxin production. This process highlights the interconnectedness of the microbial world and its impact on bacterial evolution. Section 4. Transformation. Picking up genes from the environment. Bacteria can also acquire new genes directly from their surroundings through transformation. Imagine a bacterium swimming in its environment, encountering free-floating DNA fragments. These fragments might come from dead bacteria that have released their genetic material. The bacterium can take up these DNA fragments and incorporate them into its own genome. It's like finding a discarded instruction manual and adding some of its instructions to your own book. This new genetic information can provide the bacterium with new traits, such as antibiotic resistance. Transformation demonstrates the remarkable ability of bacteria to adapt and evolve by utilizing available genetic resources in their environment. Section 5. The importance of understanding bacterial gene transfer. Bacteria, despite their microscopic size, possess incredible mechanisms for sharing genetic information. Conjugation, transduction, and transformation are powerful tools that contribute to bacterial adaptability and evolution. Understanding these processes is crucial for combating bacterial infections and developing new antibiotics. By studying how bacteria share genes, we can develop strategies to prevent the spread of antibiotic resistance and stay one step ahead in the fight against bacterial diseases.